Hey guys, I don't know that I can bear to be on camera again until I have to do Carol. I'm just kidding. So camera weary, but I'm assuming you can hear me and see the babies. Dr. Tudge said he would come back in if there's a lot of people in here. I see there's 7 or either they just left the tab open. Oh, here he comes.
I'm back.
Thank you. You don't need me on camera, right?
No, not at all. Not at all. Everyone wants to look at your babies anyway, Mary.
I'll leave them up. Perfect.
I'm sure they'll be ignoring me and watching your baby crabs.
I doubt that.
So what didn't we cover last time? Did anyone have any specific questions that we can get through in this session at all?
I'm scrolling back up. I love the comment that Kelsey did that's pinned.
Oh, yeah. Okay. Absolutely. I mean, there should be more of it, but usually most scientists are a little bit toffee-nosed about hanging out with non-scientists. So I'm not, but many are. But
well,
I mean,
really
there's a huge movement called the citizen science movement, which of course is all over all the areas of research, archaeology and all the areas of science, bringing citizens in to gather data. So this really is the same sort of thing.
Yeah, like meteorologists who rely on people in certain areas where they can't be.
Yeah, correct. Yeah. Yeah, I think Kelsey, you're right. It's the problem is the data is often almost like a one-way system where the citizen science gather data goes to the researchers publish it, and it never makes its way back to the people who helped gather it, because the publishing in scientific formats usually excludes it from public access. So there's a little bit. It's a little bit easier these days. A lot of people are publishing in open access and open format journals, but there's still only the stuff that scientists routinely access, and not the formats that the general public accesses. So yeah, there is a bit of a problem with getting that data back again. I mean, just thinking about those fabulous videos we just saw from Tammy's work. I mean, really, they need to be up there for everybody to see somewhere to inspire people. But I don't know, if you put them on YouTube and there's so much stuff there, it just gets lost. So I don't know what the right solution is for getting that material into faces of people who want to see it. That must be the big challenge in the information age.
Yeah, you collaborate a lot, it seems like. I mean, just from what I've seen of your papers and your projects and your lab work. Yeah, could you just say a little something about the joy of collaboration?
Yeah, I mean, I've always been a collaborator ever since I was a graduate student. I guess I got that from my PhD supervisor, Professor Jamison in Australia, and because he was an international collaborator, and I saw the types of work that he did with other people. I saw him working with people all over the world, and I guess I just picked that up. And I've worked with people who are not good collaborators, who are very proprietary about their research and their work. And I think to myself, it's so counterintuitive. It's like this work's being done to be disseminated, and it should be disseminated as widely as possible. And people should be combining their skill sets, etc. But there are some people who their own advancement and their own their own personal value needs to increase, and it usually happens faster if you're a little selfish. So that's often what happens in science. People keep stuff to their chest so that they can be the next person to get the next cool paper, or get promoted, or get the right job. And when really, I think collaboration is the best way for disseminating research.
Yeah, I mean, it's how science works, kind of,
yeah,
right?
Exactly. Yeah. Kelsey, you said the big something of the big crab sites like CSJ. What is that? The only CSJ I know is the Carcinological Society of Japan. What am I missing there? Oh, this Crabseed Journal, of course. You guys all know that acronym so well. Yeah, sorry, my bad. I do know that. I met that many years ago. In fact, I think I had some T-shirts printed when I was a new postdoc at the Smithsonian in 1995, and I think I got it through some site or access through the Crab Street Journal, if I remember at the time. So yes, well known. Yeah, you're right, Robin, about access. That's the problem. In many cases, most journals are being run by private societies or commercial publishers, and so access means money to them. We're trying to move away from that a little bit, but there's only a very limited field of open access or freely available scientific formats, because of course somebody has to make money out of it somewhere. So it's becoming more popular, but often the payment has to be up front. Often, you may not know it, but the scientists actually pay to have their material open access for everybody to access. And that way the publisher does get paid irrespective of how many people access that. But it's a slowly evolving model. The publishers, of course, were all print publishers for a long time, and now they're moving to electronic format, and they're still trying to recoup the costs of not printing textbooks and selling hard copy journals and reprints and things like that anymore. So it's an evolving model, and unfortunately, it's still run by economics of publishing. But I think it will change. But for you guys, it has to be frustrating. I mean, I can't access all journals. Sometimes I have to pay as well. But my university has library pays these publishers to access as many journals as they can. So yeah, it can be frustrating. There's a few. I know of a few sites my students tell me where you can access stuff for free. There's a whole lot of pirated papers you can access through a couple of Russian sites. But I always tell my students to be so careful going through Russian websites to access papers. But there are some places you can find this for free. There's a few groups who have put a lot of it together and made PDFs and made them available. But if there's ever anything you really want and you can't get anywhere else, just let me know and I'll see if I can access it. I'd be happy to do that. If it's just a PDF, it's just 60 seconds worth of work to download it, copy it, and send it on to you. So yeah, do let me know.
That would be awesome. Likewise, I will extend the invitation, because I know Stacy means it, that, if there's something that comes up for you that you think the crazy at-home crab researchers can help you with,
yeah.
we would rush to do it.
Okay. Thank you. Yeah.
I do have some fairly small perlatus that I got earlier this year. They're still in the white sort of candy stripe phase. So I've just let them be. They're fairly new. I was letting them de-stress in their new home. But I'll sign up to see if I can sex them and check them after molts and see if I see any sex tubes or any change in sex tubes or anything like that.
Yeah, okay. That would be interesting. Like I said, I don't know when that transition is. We don't know when that transition is. If it's sexually related, then it should start as soon as they are competitive for access for mating. But no one really knows. So that would be interesting information to get. Even just recording if they're ever trying to get involved in reproductive encounters or chasing other crabs around for that reason, or whether they hang around larger males who are involved in reproduction. Even that sort of information would be useful to see what they're up to. And I was thinking about the question earlier about how do we see and measure these parts on these crabs that are going to, the crabs that definitely don't want you to do it, so they're not going to be cooperative. And the only thing I could think about was it would be interesting if you had them on a mirrored surface, whether you could get them to come out and you'd be able to see in the mirror underneath, but I don't really know how that might work. But that's something you might think about because they may try to come out of their shell more so when they're facing down on a substrate. And if you had something like a mirror underneath that you might be able to angle to see. But I really, I don't know how impractical that is because I don't handle these crabs on a daily basis like you guys do.
Right. It'd be a lot easier if we were decapods and we could, have 10 hands to.
There you go.
For some listening, I have found some success because we're trying to sex the babies as soon as we can tell. They're just so small that sometimes you can't see any gonopores or anything. But I've had success, and others have had success for the people who are listening with if you have a glass lid on your tank, close the lid, hold them above it so they can see the substrate below, and they can just feel the glass, but they can't get a grip. They can't grasp anything. And they'll often keep stretching, and you can raise the shell and they'll keep stretching. If they're tiny, you almost need someone there with a camera to capture it because it's hard to see it in real time. But that's one way that's been helpful for us.
You could even do it on your phone screen and take a picture while they're stretched out. Although I don't know if it'll. Of course, the lens is not the lens is not where the screen is, so that would be difficult. Well, you might think about trying to do that. I don't know if the camera can, the phone can focus at that distance. But I was just thinking about that while you were talking.
Yeah, that's a cool idea. I mean, we try everything. Moa's our best, one of our best photographers. She's got some amazing close-ups of the compound eyes, and I don't think you may not have seen the Variabilis talk, but she had just incredible footage too. Really, just beautiful close-ups of their structures, and she's trying to raise them too. Helena Gwoli, she's in Australia.
Oh, okay. Yeah, being an Australian, I was interested in knowing what whether people were raising Variabilis outside of Australia or not. Is this something that you guys have access to, or is it only something the Australians have access to?
No, we have it as I can give. Once we get all this stuff downloaded and moved to Patreon, we will definitely give you the link to get in there and look through whatever you want to look through, to review anything. They're very involved with us. Sue Brown actually read Variabilis. She's also in Australia. Helena Gwoli has come close. I think she got them to Megalopa. So yeah, anything we've got out of this conference too is yours once we get it organized. Hopin gives us links to download all the chats, all the sessions, all the main stage talks, everything. So we'll be laboriously moving that over in the coming weeks, and hopefully we'll have all of this, kept for posterity.
Yeah. I was actually thinking more along the lines of the actual crabs themselves. Do you guys have access to Variabilis? Are they coming out of Australia or not?
No, not at all.
I didn't think so, because about maybe 7 or 8 years ago, I actually got a request from the Australian government to review a permit to export Variabilis, I think it was, or maybe it was to import other land hermit crabs into Australia. And I was asked to assess it and provide my recommendations for part of a permitting process. And I couldn't remember which way it was, whether it was Variabilis out or whether it was other exotics in. And I basically, cautioned against it for a couple of reasons. One, if you're a country that has your own species of land hermit crabs, I'd be worried about introducing exotic parasites and diseases and stuff to wild populations. But I also was concerned about the global exploitation of wild populations, and so I generally was negative in regard to that. But I was curious because the Australian government is usually fairly strict with its import and export, and we have a much, much stricter pet trade in Australia than we do in other parts of the world. So I was just curious about that fact.
Yeah, no, I think that's smart of them. I lived in Hawaii for quite a few years, and they had the same after having awful things happen. If you ever know anybody, so I think that worry is on Australia's part is good for incoming. as far as coming out, if you know of anyone who can get permits for research, for study, for whatever, I would love to talk to that person if it would ever be possible to get them in this country, or purpurious too. Those are also not coming out in any way. And I don't mean to sell, I mean for raising, for the educational purpose of it. If that ever comes up or becomes a possibility, please think of me or our community or some of the people that are doing this.
Certainly. Yeah, I don't know. I saw several of the speakers. I saw that Sue and the other woman were probably Australians, and so I knew that there was a community there. But I never interacted with that community when I was there. But I've been in the States for 20 years, so I was a young, postgraduate researcher back then. So I didn't really have the connections I have today.
Perfect.
I just saw a question from Robin Louie asking where exactly those sexual tubes are. Yeah, they're always on the base of the last set of walking legs, Robin. So what we call P5, peroipod 5. So the keelers are one. Then you've got two large walking legs that appear out of the shell. And then you've got two small legs, which are not really walking legs because they're only inside the shell. And they're used to grip the inside of the shell. And so that last pair is the fifth pair. Right on the base of them, the basal segment is called the coxa. That's where you'll find sexual tubes if they have them, and they should be the male gonopores paired usually are in that position. Basically, the hardest part to see, they have to get their entire ventral surface out of a shell, and you nearly have to see exposed soft abdomen before you can actually see the P5. It's a little easier for females. You only have to go to the base of the third legs, but it really is one of the hardest places to get access to visualize stuff without the crab coming completely out of the shell. I can remember trying many different techniques to get hermit crabs out of shells when I was doing my PhD research, and there were so many different. And I wanted to get them out. I didn't want to save the shell, and ultimately I was sacrificing the crabs, but I wanted them out alive. And there was many techniques that I was told over the years. Heating a shell to make a crab leave it, cutting the tip of the spire off with a pair of pliers and poking something like grass or wire down there, and things like that. All sorts of strange techniques. And I found for some crabs, all you have to do is hold them upside down and they drop out. And others, you could apply a gentle heat to the spire of the shell and they would just crawl back inside and die. So most of them were not useful techniques at all.
Yeah, that abandoning the shell is a pretty common thing with compressus, and that was what led us to think there was the slightest possibility that we might have a hybrid, because a bunch of the 2018, a percentage of the 2018 babies were very happy abandoning their shells. Compressus, because they walk so much faster, have much lighter shells, right, in the wild. And apparently it will abandon them in the wild. So that was one of the behavioral things. But yeah, nothing confirmed. Just weird behavior that we didn't have any other way to explain.
Yeah, where do compressus naturally come from? I can't remember.
Ecuador, I guess.
Okay.
And,
okay. So they're Central American. Yeah. Are they a Pacific species only? Do you know, or are they in the Caribbean as well?
I don't know the answer to that. I think they're only Pacific. I think the only thing Caribbean is Clypeatus.
Yeah, I think so. I think Mexico and places like that have compressus on their coastlines too. Costa Rica and places like that.
Yeah.
So it might be a Pacific species. No, that's interesting that they have a very different behavior to the rest of them. And their access to lighter shells might simply be whatever species of mollusks are living in the surrounding waters, because that's the shells they're going to be accessing.
True. Stacy was posting pictures on our main site too, that the compressus have a much larger thorax shield, whatever, the part that goes down towards the tail. She had pictures and it's much wider, and they actually prefer, we know this as keepers, that compressus prefer the D-shaped opening shells as opposed to the O-shaped, the circular shape. Yeah.
Okay.
So. And they don't like to leave shells. They will keep a shell forever till it's the tiniest little thing on their butt, and they have to move. And then when they do move, you can see they've removed almost the entire inside of the shell, the spire, everything.
Yeah, they've been known to modify shells for sure. And
you know if they're doing that with secretions from their abdomen, because it's so smooth. It doesn't look like it's. I know they modified the top shells with their claws because we have pictures of that, but I don't know how they modify the inside, the interior.
That's really interesting. I'd only heard of them modifying the outer parts by rubbing basically their legs and chelae against it. So if they're smoothing out the inside, I don't know whether that's just wear and tear or whether they really are secreting something. If it's a secretion, they would have to have the tissues present. The telltale thing would be the presence of, say, glands or glandular tissue to secrete something. You'd have to look at the skin. You basically have to section it and look at the skin to see if they've got the glandular tissue to secrete something. But that's really interesting. I've never heard of that before.
Yeah, none of us really know why either. And I haven't seen it in my Clypeatus. Their shells look normal. It's largely compressus, the lilas. They had almost nothing left in their shell. It was like a husk, right? So that's also, maybe why those species are so much more comfortable wearing plastic debris, because they're used to wearing something that doesn't curl around their tail. But yeah, great mysteries.
It's so counterintuitive to survival. It's like wearing down the inside of your house until the walls fall down. Where's the survival value in that? What is evolution telling them to do in that regard? And is it just that shells are so scarce that they have to hold on to them for so long that it's a natural wear? Or is it really something that they're doing to make them more comfortable, to make more room in them? I hadn't really thought about that because I've never heard of that before.
Yeah, I think it's fascinating. And we've seen that those previously modified shells are a premium for when they're abandoned and a crab takes another shell. Those shells are the premium shells that the others want to. They've been like pre-modified. It's like a pair of distressed jeans, right? That is nice and soft.
Well, that's interesting. You should read Mark Laidry's paper that he wrote. I think it was 2018, early 2019. I think it came out. He was talking about sexual tubes. He called them penises, and he wrote a very sexy paper about penis length being used to hold on to hot property. So when they got good shells, they didn't want to let go of them during reproduction, and so they have these long penises to hold on to their good property. And he had a very catchy title, like I don't know, big penises for hot property or something like that. But you should look that up. Laidry's the name. I think you wrote it before. Laidre, first name Mark, and I saw him present this at a conference in 2018 here in Washington D.C. and I think it came out soon afterwards. And someone had written a reasonably popular article about it as well. So that was talking about modified shells became really hot property, and so to hang on to them, they had to modify their reproductive behavior or something. I'm unconvinced, but it is an interesting idea.
Yeah. Cool. I'll look it up. And before I do, my husband will come in here and see my note. Mark Laidry, penises and shells. No, he won't. He won't wonder. He'll know.
Yeah. Just like my family is not surprised to see me sitting on the computer talking to a whole bunch of women about sex and crabs. So it's like, whatever. Yeah.
Does anybody else have any questions? I see Kelsey and Moa are answering, and keeping up with us. We've only got 6 in here. So, so if you guys have questions, definitely. I love that you're listening to and contributing.
And remember, as I said before, if you have a particular topic that you think you might have some data to address, then contact me offline, send me an email. I gave it to you before. I'll post it again now. It's just ctudge@american.edu. And happy to explore it with you. You know, you can show me a little bit of your data, tell me what you think you want to use it for, tell me how you got it, and I can talk to you a little bit more about maybe changing methodology for data collection or whatever. And we can just start to investigate these issues and see if any of it's useful.
That'd be wonderful. Yeah, that'd be great. I think MOA needs to have the Lundberg method for identifying crabs by CV patterns. But
there you go. That's if she's got the data, I think she has. She probably has nearly all of it already. So I'd be very happy to chat with you, Mo. Send me an email and let's start to talk about that and see what can happen. And like awesome. I said, a lot of you probably have this data already. You just haven't probably been collecting it in a way that's going to make it immediately useful for research. You just have to think about modifying the way you collect data. You have to document everything. You have to think about the concept I mentioned before of a control. Read up on the scientific method. That'll take you back to your high school biology classes. But if you're thinking about things like that, think about controls, think about variables. If you're interested in one variable, you have to control for the others. Just that sort of way. And I'm happy to help you to work through those things. You just have to remind me who you are when you send me an email. We can slowly work on some of these things. And I'm sure we can get some great data out of you guys, because you have more crabs, more data points than any researcher who's currently doing any hermit crab research in the world right now. Your community has that data. We just have to get it out.
Yeah, I don't even know if mine is useful, but it's very useful to me. The first time I actually brought them to land, that was all I could do. But as I got better in my methods, I've been keeping numbers. I'm really, really interested in the numbers, like the numbers of stage, how they change, from stage 1 to stage 2, how the population decreases, how long it takes for them to get to land, like all of that. And I've graphed some of that. So as soon as I can collect myself, I'll send you some of that and you see if you think anything in that is useful.
Yeah, no, absolutely. It'll be fun to look at. And as I said, there's very little data on this. For somebody to do that for a wild population, they have to go out and they have to use plankton nets and they have to sample the waters around, for example, an island where a species lives, and they have to sample it for the several weeks that those larvae are in the water column. And they have to quantify and count it and sort it out from the millions of other plankton that's there. And quantify the numbers and the sizes and to catch all that. It's just a tremendous research undertaking. And although it's because it's in captivity, you can't directly apply it to the wild, but it is really useful information and it can be obtained so much easier than anybody who has to go out and do it in the wild.
Right. Yeah, it's one data point, but it's still interesting, right? Yeah. When you're giving them everything you think they need and taking away all the variables of predators, how many survive?
Yeah, and I think when you and somebody asked a question about whether they thought your survival rate was better, I think it probably is better than what's happening in the wild. You said 390 from 400 from 10,000, but you think about how many mature female crabs are dropping 10,000 eggs off in the ocean every season, and you're probably only getting hundreds of or even thousands of crabs walking themselves back up onto those beaches later. So I suspect you have my intuition tells me you have a much higher survival rate than anything in the wild. Yeah,
it's very artificial and skewed towards success as much as I can.
Well, those poor wild larvae don't have you looking after them, Mary. Right? So if they did, they would do so much better.
Oh, you're funny.
Yeah.
I think we may be done. I don't want to keep you too long. I've got if you're interested, at 3 o'clock, Carol Ann Orms, the woman who has the 44-year-old, almost 44-year-old in captivity hermit crab, she's 83, is going to be on screen with me, showing off Jonathan, talking about how she's kept him over the years. And she doesn't have a talk planned, but I was just going to talk with her and get, get some information, and people can ask any questions in there. You may not want to stay the whole time, but it might be useful to see what
Okay, so that's 3 p.m. Is that a session or a stage?
It's a session. We initially wanted to do the stage. It's a little hard for her, a little harder for her, I think, to do that, and for me as well to navigate the stage. So we're going to do a session because it's a little easier, like you and I are doing now. And I'll post the link on the main chat to encourage people to go to that session room shortly.
Okay, that sounds good. Well, thank you, everybody who came back again. I'm really impressed to see that you guys are interested in this. And as somebody just said, the data that collected can only be better for raising and keeping your crabs and your animals. So I'm happy to participate and see what we can get done. Don't be afraid to send out any messages to me. Drop me emails. Like I said, just remind me who you are and where we met, and we can start working on that. And don't be surprised if you don't get an answer in 24 hours. I will get to it eventually. I tell all my students that I'm not carrying my phone around, reading all my emails, and I usually on a daily basis check in at the end of each day, depending on what else I'm doing. So be patient. I'll get back to you in a day or two if you send me something. And I'm happy to interact with any of you. And let's keep in touch. Same for you, Mary. Let me know what's going on. And if I can be of any help to anybody in the community, then happy to do so.
Fantastic. Thank you. And hopefully we'll meet on Radiolab at some point, too.
There you go. I'm still waiting to hear about that. We'll see what happens. It'll be fun.
Yeah.
Rachel said she was going to try and drop into this. I don't know if she did or not. I told her about it, and she went and looked up on the web. She said, "Oh, that looks so cool." So I wasn't sure if she was going to try and participate or not. But that would be it's going to be fun to hear that when it comes around.
Absolutely, I can't wait. Thanks a
lot. My wife said she's going to start knitting more hermit crabs, and she thought she might be able to sell some to you guys at some point. And I said, "You bet." They're probably all crazy for them.
She could take orders and fill them, and then she'd know how many to make. Although she might have an order for 10,000. But
that's what I said to her. I said she should be careful what she bites off here. She may not be able to chew it. So we'll see what happens with that. Yeah. All right. Well, I am going to check out now. I might drop into a few other sessions and see stuff. It was great to interact with you all, and please keep in touch.
Fantastic. Thank you.
All right then. Bye, y'all.